Hello everyone, my name is Ronnie. This is NBB 299 and I'm here to talk to you about my research. A lot of it is centered around this condition I have called Tourette syndrome. My medical condition has shaped a huge chunk of my journey, and it's also the reason why I can't talk properly unless I have this stick in my mouth. So just bear with me on that. Before we get going, I really encourage you to give me feedback. If you have the slides, that's the link right there. But if you don't, there's going to be a QR code in the corner of these slides. So just scan it with your phone camera and it should link you to the form. Feedback is always super important to me because it tells me how I can improve. So hi, I'm Ronnie and I'm a grad student in the neuroscience program here at Emory. My research is not solidified yet, but I'm slowly transitioning to do more work in biostatistics and bioinformatics. My goal is to have a project that combines these areas with neuroscience. I got my BS in neuroscience at Brown. I was born in China, moved to New York when I was 1, and raised there my whole life. Then went to college in Providence, and now I'm here in Atlanta. I have a lot of weird non-science artsy hobbies like drawing and writing. I love memes, and I have an online portfolio that you're welcome to check out here. Okay, so like I said, I have Tourette's. It's a neurological disorder that makes me twitch and make weird noises. I do have a symptom called coprolalia, which is the involuntary use of bad words. But thanks to #technology, hopefully my tics won't be an issue. Being diagnosed with Tourette's really motivated me to study neuroscience, because in a way it was like going on a journey to learn more about myself. Neuroscience also appealed to me because we literally are our brains. When you hold a human heart, you might ask, whose heart is this? But when you hold a human brain, you don't really ask, whose brain is this, do you? No, you ask, who was this? It's different. It's more meaningful, at least to me. And there's this weird feeling when you look at a brain under a microscope and you realize you're using the same exact thing that you're studying. My journey in research started at Yale in the summer of 2010, right after my junior year in high school. I did a research project on Tourette's, and that's the one I'll be focusing on today. But after that, I was hooked, and I spent a lot of my time in college and now grad school doing research on things like Alzheimer's and reinforcement learning. The title of my research project was called Decreased Density of Nose Interneurons in the Striatum of Individuals with Tourette Syndrome. If that doesn't make sense, don't worry, it's not supposed to yet. We'll get there, trust the process. So here's some background. There's this collection of structures in the brain called the basal ganglia, and it's super important for motor control. Dysfunctions in this really complex basal ganglia circuit have been linked to movement disorders like Parkinson's and Huntington's. In vocab time, the striatum is this structure circled in yellow, and it's made up of the caudate and the putamen. And almost all input to the basal ganglia goes through the striatum. So it's very important. Okay, say it with me. Striatum equals caudate plus putamen. So I made this disgusting slide in high school when PowerPoint was still somewhat of a miracle. It just shows that an interneuron are a type of neuron or brain cell, and they're really important for integrating and processing all kinds of information. We know that there's this type of interneuron called the NOS interneuron. NOS, N-O-S, stands for nitric oxide synthase. It's just a protein that this type of cell has. The NOS interneuron is GABAergic, meaning it's inhibitory. Previous studies have shown that patients with Tourette's had abnormal levels of other inhibitory interneurons that expressed PV, SST, and NPY in the striatum. Additionally, destroying nose and cholinergic interneurons in the mouse striatum leads them to have these weird, abnormal turning behaviors. So naturally, all of this evidence is begging us to investigate these nose cells next. It might not be surprising then that we thought nose interneurons would be less abundant, like everything else, in the striatum of TS brains compared to controls. Again, striatum equals caudate plus putamen. And now that we have this hypothesis, how are we going to visualize and then count all those cells? Well, I'm glad I asked. We use something called immunohistochemistry. Basically, we take very thin slices of preserved brains of people who had died, and we dumped a bunch of chemicals in a very specific order onto them. The basic idea was to get an antibody to bind to NOS. Remember, NOS is just a protein. Then we add a secondary antibody to bind to the first one. And then we add this weird, colorful thing on the right called an ABC complex. It's just a big chemical structure that we use so we can amplify the signal and make it more detectable. And finally, we add a final chemical called DAB that reacts with those red boxes, turning all the cells brown. So now we can visualize selectively these nose interneurons under a microscope. And we did that. And we used some really expensive software to count cells. We basically counted a bunch of random representative samples, and then the software did all the estimation and extrapolation for us. So this is what we found. Control brains on the left column, Tourette brains on the right. These are pretty representative sections, and we basically saw that there were fewer nose interneurons in the striatum of TS brains, as you can see on the slide, indicated by the arrows. And then we quantify these results. Remember again that striatum equals caudate plus putamen. Well, in both areas, the TS brains on the right side had a lower density of nose interneurons than the NC, the normal controls, on the left side. The horizontal bars are the means, and this was statistically significant. But I admit at the time I was really bad at making figures, so I could barely get the p-value to show there. In conclusion, remember that these nose interneurons are GABAergic. They are inhibitory. If TS brains have fewer of these inhibitory interneurons in the striatum, whether it's NOS, whether it's PV, SST, all these things that we've found before. If TS brains had fewer inhibitory interneurons in the striatum, that would mean that there's less inhibition in that whole circuit. And that could explain the excess unwanted movements we see in Tourette's. Of course, my sample size was really small, with 5 people in each group. And unfortunately, that was the best we could do because it's really hard to get Tourette brains. This deficit in so many various types of inhibitory interneurons suggests that maybe we could increase GABA levels in the brain, and maybe that would help with some of the symptoms. There have been attempts to supplement GABA, but so far they haven't been too successful because there's actually a lot more going on than just GABA. For example, dopamine is also huge, and we think it's really important in Tourette's because for many people, drugs that block dopamine can actually help control their tics. That study was very small, and it seemed very insignificant at the time. But it actually got me a lot of recognition and was even incorporated into a publication. So one valuable lesson I learned along the way was to never discount the potential importance of your work and your research. Even if it seems trivial and tedious at first when you're doing it, you really, really never know where it might lead. Honestly, the present and the future are pretty scary for me. I think I've come pretty far in my neuroscience journey, and I've decided to transition into biostats and bioinformatics for several reasons. One, I currently suck at math and I want to develop my quantitative skills. Two, I think the world is moving in the direction of big data, and so having these skills will be helpful for my professional life in the future. And three, while I love neuroscience, it's just not really practical for me to have severe tics and handle things like animal surgeries and these dangerous chemicals. So while I don't have a clearly defined project right now, a thesis project right now, my goal is to try to connect the fields of neuroscience and data science, maybe through an intermediary like psychiatry. And eventually, I hope I can continue doing research and fulfilling my passion for teaching and advocacy, whether it's in academia or elsewhere. Through everything, my journey has taught me some really important lessons. I've come to appreciate how hard work and perseverance can go a long way. My magic number is 147 because during my junior year in high school, it took me 147 emails to secure that research opportunity at Yale. Despite having this really stigmatized condition, I try to let it be more of a motivator rather than an impediment. And here on the screen are two quotes that I found on the internet that have stuck with me for years and years. With that, I want to welcome you on board the struggle bus. I want to remind you to please give me feedback through that link or the QR code. My contact information is on the bottom of the slide, and you are always welcome to get in touch. Thank you all so much for listening. I hope my talk has been somewhat informative, and I look forward to this pandemic ending and then to hearing about all of your successes and scientific achievements in the future. Thank you for having me. Have a great day.